morning, everyone. Good afternoon. I'm Cassiana Montagner. I'm professor at University Institute of Chemistry of the State University of Campinas in Brazil. And it's a big pleasure to be the moderator of the session 2, Team 5, about priority emergent pollutants in the hydrocycle, and talk about microplastics, nanomaterials, PFAS, and PPCPs, together with the co-moderator, Robert DiFilippo, that is here with me. He is lecturer in the hydrology at the National Institute of Geological Science at the University of Philippines, and also Piero Gardinelli from Florida International University in US. So welcome to this session. Before it starts, you may know, but it's nice to remember that to all of the speakers, that this session has been recorded, and for all of you, we invited you to submit your questions in English via QA button. You can see this in the bottom of your screen. We will address your questions as much as possible after all of the talking, and also writing the answers during the session. As we have a huge audience, if you don't have time to answer your questions, we will receive over, and then we'll collect all of the responses from the presenters, and then publish it in our website. Please note that the chat box is displayed during the session. However, we ask to keep your eyes open to that, because could have some useful messages that share during this the presentation of your session. You also have the options to switch on the automatic subtitle at the bottom of your screen if you have difficulty to hear. If you have experienced technical problems, please Google Zoom Help Center or send a message to the conference online for assistance. Then let's start it with our first presentation, that is from Hadi Toure, environmental engineer from National School of Engineers in Mali, that we present nanofiltration of perfluorooctanoic acid and perfluorooctanoic as the functions of water matrix properties. So welcome to our session. You have the word.
Hello, everyone. Thank you for having me in this session. My name is Hadi Toure. I have performed this research while studying at the University of Central Florida, and I currently work as a utility engineer and part-time lecturer at the National School of Engineers in my hometown in Mali. I'm also active in youth network in water and sanitation. Today, I will discuss about the solutions that we investigated for the removal of perfluorinated compounds, commonly known as PFAS, in water. Next slide, please. PFAS are man-made chemicals manufactured in consumer products such as food packaging, non-stick cookware, also firefighting foams. They have a special carbon-fluorine bond that grants them bioaccumulative properties and makes them very persistent in the environment. The diagram on the right shows the chemical classification of PFAS. The products investigated in my research are PFOS, which stands for perfluorooctane sulfonate, and PFOA, which stands for perfluorooctanoic acid. And these are the two main and commonly investigated PFAS. PFOA is used in firefighting foams and therefore has a foamy texture, whereas PFOS has a sulfonic group that makes it sticky, and it is used for its adhesive properties, like in Teflon, for example. Next slide, please. So when we investigated these PFAS in 2018, they were found in both surface and groundwater in several locations in the United States. The diagram on the bottom left portrays the different pathways in which these chemicals contaminate our drinking water, from industrial discharge points to waste and stormwater and soils. On the diagram on the right, bottom corner, you can see that these chemicals have serious suspected health effects, especially on women. Next slide. It is commonly known that conventional treatment systems are not effective for the removal of PFAS. The removal methods stated here are classified in order of efficiency. We can see in some studies that granular activated carbon have a removal efficiency of around 80%. Ion exchange was shown to have a slightly higher removal percentage. And of course, membrane treatment is the most efficient method to remove these chemicals from our water. Next slide. So we have chosen nanofiltration as a treatment method in order to investigate the behavior of the PFAS around the membrane surface. By nanofiltration, there are two main rejection mechanisms that were identified in literature. First, you have the size exclusion, in which bigger molecules are strained from the membrane surface, and electrostatic repulsion. In the literature review, we also found that very few studies use real water matrices. And finally, PFOS rejection was increased in the presence of calcium and magnesium cations. Next slide. Based on these findings, our objective was to first investigate the rejection of PFOS and PFOA using nanofiltration, and second, to determine the behavior of these PFAS in laboratory prepared water, surface water, and groundwater. This table shows the characteristics of the different water used, and we can observe that groundwater has the highest amount of calcium and TDS, which stands for total dissolved solids. Next slide. So how did we conduct these experiments? We used a laboratory bench scale membrane setup that you can see on the bottom right quadrant on the picture. The membrane material was a flat sheet polyamide thin film composite membrane. In this setup, we spiked a known concentration of a mixture of PFOA and PFOS and collected the raw and filtered water at time t=0 and then after 20 hours. Then we extracted the chemicals using solid phase extraction and analyzed the samples in a liquid chromatography mass spectrometry instrument called the LCMS. It is important to highlight that because these PFAS are everywhere, we had to order special materials in the tubings and samplings to avoid cross-contamination. Next slide. From the results, we can see on the diagram on the left that compound rejection was greater in real water matrices in groundwater and surface or river water, and the rejection was greater in groundwater, especially for PFOS. On the right, you can see the picture of the used membrane flash sheet after the 3 experiments conducted. Next slide. Can you go to the next slide? Okay. So size exclusion plays an important role in the nanofiltration of PFAS. The presence of sulfonic groups in the molecule of PFOS may favor PFOS cation complexion. So the PFOS molecules will bind with magnesium or calcium and form a bigger molecule that is better rejected by the membrane surface. Also, cations and cations, such as calcium and magnesium, might act as a bridging agent between 2 PFOS molecules resulting in higher rejection. And finally, organic matter combined with cations may result in forming a fouling layer on the membrane surface, which can also enhance rejection and the flux decline of the membrane. Next slide. So I would like to conclude with what I'm doing with these results after moving back to Mali. In the opening session of this conference, it was mentioned that there is very limited understanding of scientific knowledge of these emerging pollutants. As far as we are concerned here in Mali, there's no PFAS investigation to my knowledge, and very few in neighboring countries in West Africa. However, we do use plastic, the same imported food packaging, non-stick cookware, detergents in our households. On top of investigating the occurrence of these chemicals in our drinking water in Mali, I want to try to directly apply scientific knowledge to raising awareness sessions to not only young environmental engineering students, but also general users of these products. We have done movie screenings with Dark Water, which is a movie that showcases the origin of these chemicals. When I first watched that movie or heard of PFAS in US daytime television after this research, I realized how much it is important to simplify scientific jargon so that the public can relate easily. In some of the discussions with my students, we talked about turning back to our traditional pots and pans or giving up on the mentality that imported goods are better than local goods. Finally, I would like to thank the organizers of this conference, IWRA, and the inclusion efforts of UNESCO to provide and produce open source science for all. Thank you for your attention.
Thank you very much. Very interesting presentation. It's our pleasure to see you here. So I would like to invite the next speaker. That is Silvia Pedroso Melegari from Center of Marine Studies of Federal University of Parana in Brazil to present on the toxicity and biodegradation of pharmaceutical diclofenac employing the green marine microalga Tetraselmis as a preliminary study. So welcome to this session, Silvia.
Thank you, Cassiana. So it's a pleasure to be here and present some of our results of our research group. Thank you. Next, please. So as well introduced for my previous colleagues and panelists, pharmaceuticals are also viewed as a chemical that is classified as an emerging contaminant. So the most problem of this chemical is because of the conventional wastewaters cannot treat appropriately or remove these chemicals from water. And the use of photobiodegradators, next please, that employ microalgae to treat wastewaters, and in particular emerging pollutants, can be promising and recent subject that are very explored in the recent literature. So, however, understand the toxicity of the emerging pollutants over microalgae is important to understand the limitation of this technique. Once that biological processes are sensitive for toxic pollutants. Next. So with that, we have some goals with this research that are evaluate the toxicity of the sodium diclofenac to the marine microalgae Tetraselmis sp, and also evaluate the potential of this microalgae to biodegrade diclofenac. Next, please. So this study was split in 3 steps. The steps 1 and 2 were basically establish the culture of Tetraselmis sp, and the step 2 was expose the Tetraselmis to this chemical, and the step 3 was basically the biodegradation tests. The toxicity tests were based on the procedures established by the Brazilian Association Technical Standards, and the biodegradation tests were performed were based on the effective concentration 50 or EC50 that is obtained from the toxicological tests. We tested two different concentrations of diclofenac, 10 and 50 milligrams per liter, and this chemical was exposed to the microalgae in the exponential phase. Also, we tested two different controls, one, the negative control with only microalgae, and another one evaluating the photodegradation of diclofenac without the presence of microalgae. All the reactors were monitored at each 24 hours until 96 hours with measurements of diclofenac concentration and also cell density using UV visible spectrometry techniques. Next, please. Next. Can you put all the animations in the slide, please? Thank you. The results of toxicological tests showed a very consistent dose response behavior for the diclofenac in 4 independent tests. So for that, we could obtain from the acute toxicity estimated the EC50 from one of the best fit of the tests that was around 60 milligrams per liter. Next, please. Next. And also for the chronic toxicity, we could determine the concentration of lowest observed effective concentration and no observed effective concentration that were respectively 110 milligrams per liter. Next, please. For the biodegradation tests, in summary, we couldn't see any photodegradation of the diclofenac alone. And the microalgae could grow in all the concentrations tested of diclofenac. So this can showing to us that this microorganism is suitable to be used in photobioreactors to degrade this chemical in this range of concentrations. So for the reductions rate, we could see a reduction of the concentration of diclofenac in the highest concentration of tested in a range around 10 to 50%. But however, in the lowest tested concentration of diclofenac, we couldn't see any decrease of these concentrations. One of the problems here we can see that is probably because of the limit of quantification of the method that is very near from this concentration. Next. So in summary, we could see interesting results of toxicological data, and also these microalgae can be used as a potential have potential to be used even though with low efficiency. Next. Thank you. I would like to thank you for this opportunity. I would like to thank you for Federal University of Parana, Brazil, and all the government agencies, and also a special thank you for all the research group that I know that are watching here. Thank you, and I will be happy to answer any questions.
Thank you very much, Silvia. Very interesting presentation. I would like to invite now Vinicíus Diniz from the Department of Analytical Chemistry of the Institute of Chemistry at the University of Campinas, Brazil. Welcome, Vinicíus, to present the direct potable reuse at a prioritization of emergent contaminants for monitoring strategy in pilot scale advantage treatment.
Thank you, Professor Cassiana. It's a pleasure to be here today. And good morning, afternoon or night, everybody. My name is Vinicíus Diniz, and I'm here to present part of my research at Paracelsus Bioanalytical Laboratory at State University of Campinas, Brazil, under supervision of Professor Dr. Susan Hatt. The work I will present today is entitled is entitled Direct Potable Reuse, a prioritization of emerging contaminants for monitoring strategies and pilot-scale advanced treatment. Next slide, please. Well, let's start the presentation discussing the water. We know that from the beginning of human civilization, more specifically from sedentary civilization, water availability means the development of new culture. I mean, humans need water to survive, but also to develop their society. However, we are observing an increasing water scarcity worldwide due to the alteration on rainfall patterns and increasing of population. Especially here in Brazil, we observed a severe crisis in the São Paulo states in 2014, which showed the government agents the fragility of the water distribution system. Due to that, we must find alternatives to deal with the water scarcity that affects hundreds of countries worldwide. For example, they reuse water treatment plants. Next slide, please. Our research group passed the last years investigating several emerging contaminants frequently used in Brazil, and then started to quantify these contaminants in 6 Brazilian wastewater treatment plants. Here we can find 51 emerging contaminants, 45 drugs, and 6 sweeteners, with which our group has worked in recent years, which consequently allowed us to select 12 for further studies. Next slide, please. So for this work, we have initially prioritized sulfametoxazole, carbamazepine, hydrochlorothiazide, diclofenac, acetaminophen, propanolol, sucralose, saccharine, acesulfam, and caffeine. But we need to go further, and we started to monitor these 12 emerging contaminants in a non-potable reuse treatment plant in Brazil. Next slide, please. The EPAD Capivari 2 is a non-potable reuse treatment plant located in Brazil that operates using a process known as a membranobioreactor. The wastewater treatment plant receives sewage from around 200,000 inhabitants, with an average flow around 1,000 liters per hour. Here we can see a photo of the effluent. Please keep this photo in your mind. The membrane bioreactor technology effluent is of high quality, as we can see, for example, in the results of BDO, nitrogen, and phosphorus. Next slide, please. Once we had decided on the emerging contaminants to monitor, we had to select a powerful analytical method to quantify them in different water matters. So we carry out experiments using the dimensional liquid chromatograph coupled to tandem mass spectrometer. We had a column in the first dimension to clean up and concentrate on the sample without a laborious offline procedure. In the second dimension, we had an analytical column for the chromatograph separation. This procedure allowed us to reach satisfactory sensibility and a fast sample quantification, around 3.5 minutes of chromatography run. Next slide, please. As for the results, we have found 8 out of the 12 emerging contaminants in both raw sewage and plant effluent. It's important to note that 3 emerging contaminants were observed only in the raw sewage. Meanwhile, ricobendazole was not observed in neither. But we decided to continue our studies with 5 of them. Hydrochlorothiazide, saccharine, and sucralose were selected to their high concentrations. Caffeine was selected to its widespread use as an anthropogenic marker, and sulfametoxazole was selected since it's an antimicrobial. Next slide, please. So we have decided to conduct a study toward directly potable reuse. But what do we need for that? According to the World Health Organization, we need redundancy, robustness, and resilience, which means reliability. In this way, we need a multi-barrier process. So we started to quantify the 5 emerging contaminants we previously selected in the effluent of an advanced treatment process. But which process we worked with? Next slide, please. So we carried out some experiments in a pilot plant that continuously received the effluent of the membranobioreactor technology. The pilot plant was composed of a reverse osmosis membrane, the dark blue cycle, photoperoxidation, the purple cycle, and 1 fixed bed column loaded with granular activated carbon, the light blue circle. There we were able to combine all the process the way we wanted. Next slide, please. Starting with each process once, sulfametoxazole was removed to below the limit of quantification, regardless of the process we have used. In addition, we observed that reverse osmosis brought us the best results. So we combined the reverse osmosis process with the other two processes and thus achieved almost 100% of removal efficiency. Next slide, please. But we know that the majority of the indirect and direct potable reuse plants of the world are composed of more than two processes. So we combined the three available processes and achieved almost the same removal efficiency. Also, here we can see a photo of the membrane bioreactor effluent and the pilot plant effluents. We know that the visual aspect does not entirely govern the water quality. So in addition, we observed that the effluent of the pilot plant met the Brazilian guidelines for potability reuse, for the potability purposes, sorry. Next slide, please. In conclusion, we must keep in our mind that direct potable reuse, although complex, is a powerful machine against water scarcity. Still, due to the high number of chemical substances available worldwide, we must prioritize the most important one to make the monitoring process as cheap as possible. In addition, Brazil is on its first steps toward direct potable reuse, and further studies must be conducted. Finally, if I have to recommend emerging contaminants for monitoring purposes, especially here in Brazil, I would recommend caffeine and sucralose due to the facility of quantification and high concentration in the effluent. But other contaminants can be used in other places, as my colleagues have shown during this conference. Next slide, please. Last, I want to thank the Brazilian agency for the financial support, my lab partner for the kind time and cooperation, the UNICAMP for the support, and my supervisor, Professor Dr. Hadi, for her time and cooperation during the development of this research, and also the conference for the opportunity to be here today. Thank you for your attention.
Thank you, Vinicíus. Nice presentation, as always. Okay, I would like to invite the next speaker then, that is Piero Gardinali from Florida International University Institute of Environment in US, to present the prioritization of emergent pollutants used for fingerprint specific water source. Welcome, Piero.
Thank you, Cassiana. Thanks everybody to show up to the presentation. We have about 240 participants in this, which is it's a great opportunity. So I'm going to try to get today to an explanation of an emergent technique that is used to let contaminants emerge. So just bear with me because I have to go through some definitions. Next slide, please. Okay, thank you. So my first definition is water quality. So we are all in the same boat. Water quality is a definition, and it's a very strict definition. It relates to some kind of characteristic of a water. It could be potable water, reused water, swimming water, and relates to the chemical, physical, biological, and radiological characteristics of it. But it needs to be looked in the context of it measures or it should sustain the requirements of anything that lives in that water or use that water, whether it's a human or it's an organism. But more important to this topic, if you don't have a standard to use for compliance, you can do very little in trying to force people to clean the water. And that's what emergent contaminants are coming from. We don't have a whole lot of standards for them. Next slide, please. And then what I do is it's sometimes difficult to explain to. I try to get quotes from other people. Donald Rumsfeld was Secretary of Defense from the US many, many years ago. And this is what he said, we have things that we know, we call it known knowns. Things that we know that we do not know, we call them known unknowns. And then he goes to there are things that we don't know that we don't know. And that's what I'm going to try to introduce to you as the concept of non-target analysis. We are trying to look for things that we don't look that we're looking for. And then that's the main difference between looking for a specific compound, which is called target analysis, and looking for everything else without a preconception of what you're looking for. And that's the technique that I'm going to explain today on how to help the relevant chemicals to emerge. But also I want you to keep in mind that really you can only manage what you measure, right? So if you don't know that you're measuring something, it's going to be very difficult for you to try to manage it. Next slide, please. So I'm going to go through a few pictures just to give you the concept. If you were doing target analysis, is the equivalent of taking a picture of, a former president. So here's a picture of President Obama. Clearly, the target of the picture was him, with a lot of school kids in the background. Next slide, please. Non-target analysis is a little bit different. We just kind of back up a little bit and trying to capture all the information that we can in the situation. So this picture is a little different. You may not have the time to figure out everything that is in the picture, but that's exactly what we're trying to do. We're using analytical techniques to capture all this information. And if you go to the next slide, please, then now things have emerged as contaminants. So now I have a very specific information in this picture that I didn't even know that I was looking for, but it's there and it's for me to interpret and come up with some ideas of what's there and what's not there. So that's the essence of non-target analysis. Next slide, please. How do we do non-target analysis? We deviate a little bit from the typical analytical chemistry. We use high-resolution mass spectrometry. It doesn't really matter what kind of instrument you use, but you need to have enough resolution so the machine is capable of making very, very small differences in a mass. So this is an example of two compounds, carbofuran and 4-methate. Sorry if I cannot say that well. They are different formulas, they are different structures, but they do have the same molecular ion if you run them in a low resolution mass spectrometer. If you increase the resolution of this machine to 70,000, the instrument is capable of separating these two compounds in their masses, and that's allowing me to do elemental composition of each molecule that I detect. So what I'm using the mass spec now for is to generate all these chemical formulas of compounds that could be present in my sample. Next slide. And this is not an easy task. I end up with thousands and thousands of options. But then there's a mechanism and software and statistical packages that will go from the raw data through filtering, prioritization, database searching, and quality control. So I could identify every feature that is in the sample to some degree of confidence, but I can reduce the error on this measurement to get compounds that I am very, very sure that they are what I'm looking for. And I use regular mass spec and then fragmentation for it to do this query. Next slide, please. So I'm going to walk you through a couple of examples on how we apply this. So this is a system in Southeast Florida where I used to live. A lot of urban development, but these houses are still served by septic tanks. So the purple location is serving septic tanks. We do have some sewage collection. And then this area is really close to Biscayne Bay, which is our natural system. So our goal was to try to identify if the septic influence could be tracked by using non-target analysis. Next slide, please. So what we do, and this is the outcome that we get from this analysis. You get each compound that is detected in the sample. It's called a feature, and it's a dot in one of these graphs. And then we superimpose to that the concentration of these features and the compounds that we are very confident that we have a tentative identity to it. Then we use them in statistical analysis to separate sources. So that's what we did in this case. At the bottom right of the slide, you have results from principal component analysis, and they tell us what compounds are indicative of septic influence. If we plot now one of them, and this is sucralose, with the distance from the bay, we can see that we can actually identify the urban influence as we go from the west to the east in this area. Next slide, please. We took it further. We look at the Miami River, which is one of the main tributaries in this area. It does have a lot of sources of discharges to the Miami River, but also connects the Florida Everglades, which is our source of potable water in Miami, to Biscayne Bay, which is where we do all the recreation. So we use this as an example to see if there are additional sources in the river that we were not identified before. And again, this is a highly urbanized area. It's a managed watershed, so we control the water level with gates. It has failing sewage infrastructure and so on. But the important thing to us is that it discharges directly into the bay. Next slide, please. So similar information as I showed you before. These are all the features that were detected in 3 representative samples. The Everglades is on the left side, Biscayne Bay is on the east. And on this one, you can see much other than there's a lot more features in the urbanized areas than it is in the natural area where the water is taken. And again, in this one, the colors represent the intensity of these compounds. So the next step is trying to identify what we can figure out from this data that help us look at sources. Next slide, please. So this is, again, principal component analysis of all the samples, and we run these samples in two modes, in positive and negative mode in the instrument, so we can capture as much information as possible. And then, as you can see in this, when you look at the left plot, the samples from the Everglades area, they tend to group together. The samples from Biscayne Bay tend to group together, and you can see some kind of mixing line in between those two, with the exception of the purple dot, which is the Siebel Canal, which is a really small tributary to the river that is completely different. So we use this data to prioritize, first, the sites that we like to look for, and then to try to make connections between what this data is telling us and then what the traditional contaminants that you monitor will show. Next slide, please. So knowing what sources we have and the specific inputs that we have, we look at traditional things. So, for example, the Everglades water sample is described by high silica and high total organic carbon, where, sample from Biscayne Bay, it's a marine system, so it's higher in selenium, as you can see at the bottom. And then sucralose is a very good indicator of the urbanized areas that you go through. And in our case, the Siebel Canal is dominated by the presence of high phosphorus concentration. So now we can start making plans to deal with specific tributaries and not with the entire watershed. Next slide, please. So the take-home message here is we do have these new techniques that are coming along. I realize that not everybody has access to it, but you could collaborate with something that could run the samples for you and do the interpretation by yourself. So bottom line is, yes, we can fingerprint water. And then we can use that fingerprint created by the non-target analysis by prioritize either sites to look for or specific compounds that are capable of determining sources. That the NTA workflow does work if you look at a system overall. And then the last comment is always, I will always make that one. No matter how much non-target analysis you do want to link the emerging contaminants with the traditional contaminants, so you can actually advocate for better treatment and watershed control. And with that, thanks for listening to, and I'll be glad to answer any questions.
It was a wonderful presentation. Very interesting. So, move on. I would like to invite Marcela Ravanelli Martins, PhD student on technology with focus on environment of Faculty of Technology at the State University of Campinas in Brazil, to present about toxicity of ciprofloxacin through generation of the soil invertebrated. So, welcome to this session.
Thank you. It's my great honor to be here. Hello, everyone. My name is Marcela. I'm a PhD student at the State University of Campinas, same as Professor Cassiana, in São Paulo, Brazil. And I'm going to present part of my current research, which regards the toxicity of ciprofloxacin through generations of the soil invertebrate Enchytraeus crypticus. Next, please. So to begin with, why is this even a concern? Why would ciprofloxacin end up accumulating on agricultural soil? Well, first of all, this antibiotic is not fully metabolized by human and animal body, being about 70% excreted, which means that in the case of humans, this amount ends up going to wastewater treatment plants. And because of ciprofloxacin's properties, it accumulates on sewage sludge. While for farm animals, it accumulates on their feces and therefore in manure. So while the usage of sewage sludge, manure, and other sustainable alternatives to mineral fertilization is very much encouraged as they are sustainable, and because of their benefits to soil quality, crop production, and also their financial advantages, they might contain a few antibiotics, such as ciprofloxacin. And this antibiotic in particular, considering its physical-chemical properties, as you can see here, the KOC and half-life values, it has really high absorption on soil and organic matter, and it tends to be immobile and accumulate for many months or even years. So that's why this is a concern, and that's why in 2009, the EPA classified ciprofloxacin as a contaminant of emerging concern. So if we intend to continue with such sustainable practices, which I think we do, we must understand the effects of this antibiotic on this matrix, considering the long-term exposure. Next slide, please. Focusing on that, I exposed 3 generations of Ecrypticus in a row to ciprofloxacin in different concentrations and using artificial tropical soil. And of course, all the requirements established by OECD. After the tests were done, the statistical results showed a normal distribution through Shapiro-Wilk test for all generations, which I called F1, the first generation, F2, second generation, and F3, the third generation. And then ANOVA showed that for the first and second generation, there were no significant differences between the treatments and the control, but there were for the third generation, as you can see the p-values on the bottom. Next, please. And here we see that although there were no proven differences through ANOVA between the treatments and the control for the first and the second generations, F1 and F2, the data varied a lot with no pattern at all, such as the decline of reproduction with the increase on ciprofloxacin concentration, for example, no, which is interesting, but for the third generation, the number of juveniles declined almost the same for all treatments. This leads us to conclude that considering the long-term exposure, ciprofloxacin is likely to be toxic to this specific organism reproduction. So these results, they emphasize that the effect of this antibiotic must be evaluated with different organisms and different types of soil, but always considering the long-term exposure. So we can truly understand how badly it may affect the organisms and their primordial functions, like organic matter degradation, for example, and therefore how badly, thank you, it may affect soil and crop quality. Finally, I would like to highlight how urgent this is, as, like I said, ciprofloxacin is expected to accumulate and be immobile in soil. So if there is no established limit for ciprofloxacin in soil, which is the case, it will just keep accumulating, potentially damaging soil and crop quality and its organisms, and even enter the food chain. Next slide, please. So that's it. If anyone would like to contact me, here's my contact information. Thank you, IWRA and UNESCO for the opportunity, CAPS for the financial support, and thank you all very much for the attention.
Thank you, Marcela. Nice to see even work from our colleagues that's around us, and you don't have so much opportunity to know better. I think I also, as a Brazilian and moderator of this session, there are many Brazilians in this session and also in the other sessions also participating of this conference. We receive many abstracts and participation from Brazilians. So it's nice to see that we are engaged on this topic in Brazil. It's so hard to discuss this or have some opportunity to discuss this. So it's nice to have this as a place to think about emergent contaminants and all of the consequences about this in Brazil also, right? Okay, let's invite to the next one. It's Juan Pablo Garcia Montealegre from University of Alcalá in Spain, to present this investigating in the infiltration of pharmaceutical and transformation products through agricultural sources. So welcome to this session, Juan Pablo.
Thank you. Good afternoon, everyone. I'm glad to show you a brief presentation of a project conducted at the University of Alcalá in collaboration with the IMDEA Agua Institute in Madrid. In this project, we're investigated the infiltration of some pharmaceuticals and transformation products through agricultural soils. Please, next slide. Well, first, I'm going to introduce you why and how this study was conducted. And later, I will spend most of the time of the presentation discussing the result of this study and the conclusions derived from it. Well, for this next slide, please. For these pharmaceuticals, certain physicochemicals, certain physicochemicals properties were collected for further analysis. First, to put you in context, we should explain what emerging pollutant is. It is pollutants that are newly detected, but it's because the analytical techniques are being improved so they can reach lower concentrations than they could do it before. So the objective of this project was to investigate the natural attenuation processes for pharmaceuticals and transformation products during the infiltration through an agricultural soil from data obtained in a column test. This is because these compounds are nowadays using more and more, and they are introduced more and more into the agroecosystems as a consequence of indirect reuse. Next slide, please. So, in the physicochemical properties collected, we try to use it to explain the behavior of the compounds. For example, the molecular mass, the charge, or the octanol water coefficient were collected. So with these properties, for example, the later one, the octanol water coefficient, it is an indicative of the sorption potential of a compound, especially for organic phases. In this test, the compounds were injected at concentrations of approximately 100 micrograms per liter, and measurements were taken in the effluents throughout the test. And finally, the content of these compounds in the soil was measured at the end of the test, which lasts 19 days. On the other hand, the soil came from a corn farm in the municipality of Cienpozuelos, next to the Jarama River in Spain. Next slide, please. For the simulation, we used several models. So in figure 3 and 4, you can see the mobile-in-mobile water model and the mobile-in-mobile water model with one kinetic side. Here you can see how the interaction of different phases or regions were modeled. For example, one model can assume that there is an immobile zone or not, or in one region, there are sorption that wasn't in equilibrium, or also call it kinetic sorption. Next slide, please. The modeling was carried out in 3 stages: a stage 0 or pre-training of the data, a stage 1 where the conservative transport model corresponding to the tracer, in this case chloride, was calibrated, and an stage 2 where the reactive transport corresponding to the pharmaceuticals and transformations products were modeled. You can see in the slide some parameters and the distribution of some model or submodels that we used. Therefore, for the different models, the determination coefficient were calculated with respect of the first 5 days as a evaluation of, as a criteria for the evaluation of the simulations. This is because, as you can see in figure 5, there is flows that shows you the flow versus time. It shows that the variability of the flow measurements, however, where determined there is a lot of variability, but in this case we use a pretreatment in the pretreatment stage by means of robust and rigorous statistical criteria. We determine that the first 5 days of the test, the flow was constant. Next slide, please. So here you can see the outflow normalized concentration, and here is shown that the tracer curve is fully developed in the 5-day interval. The tracer curve is the black one, the chloride. So the parameters calibrated for the conservative transfer are not subject to the flow variability. In contrast, athenolic acid fails to fully develop the equilibrium within the first days. Therefore, the 5-day adjustment criteria may not be sufficient in this case. Next slide, please. Well, in stage 1, it was found that the mobile-in-mobile water model is the most suitable to represent the conservative transport, since it allows to reproduce the asymmetry known as tailing and even manage for obtain and determination coefficient of 1. Apart from the higher porosity of the feed, it is important to note that for the dual porosity model or mobile-in-mobile water model, the longitudinal dispersivity determined is half that of the equilibrium model, even though the mobile-in-mobile water content is only the 4%. Next slide, please. The following table shows the result of the calibrated and calculated parameters of each compound and each model. The first thing that stands out is that all the models obtained a coefficient of determination with respect of the first 5 days that is higher than 90. However, the pharmaceuticals were the compounds with the lowest sorption delay in the mobile zone, and consequently, they have the lowest distribution coefficients. On the other hand, valsartan always had the lowest degradation rate values. We can also observe that sulfamethoxazole and valsartan, the fastest compounds, obtain results that seems to indicate that the sorption kinetic behavior is negligible, since the fraction of sites subject to sorption at equilibrium was higher than 95%, and the mass transfer rate was very low compared to the transformation products. The case of the athenolic acid is highlighted because after the simulation, which we changing the weighted importance of the evaluation criteria, seems to indicate that for both transformation products, the kinetic sorption occurs at the same sites. For example, particles of certain material or size. Next slide, please. Here, there is the simulations of the model pharmaceuticals, valsartan and sulfamethoxazole, assuming equilibrium sorption and kinetic sorption, and linear and Freundlich sorption. In the case of valsartan, it is noted that it shows relative concentrations about 1, which could be due to the fact that the sorption processes are developing faster and faster, thus generating a reconcentration effect of the compound at the effluent. Sulfamethoxazole appears to show an increase in degradation, which some authors suggest may be due to for change in nitrate reducing conditions. However, it is noted that this process is reversible in nature. Also, for both model pharmaceuticals, as could be anticipated from the calibrated parameters, the model assuming kinetic sorption does not show considerable differences to the equilibrium model. It is also shown that the models assuming no linear sorption are better at representing the sigmoidal section of the step curve for each pharmaceutical. On the other hand, the transformations products are best represented by a kinetic model, whereas both models seems to show that sorption tends to linearity. In the simulation case for ethanolic acid, it was found that possibly for different initially obtained reactive transport parameters. The test data can be simulated globally. Finally, next slide, please. Oh, sorry, next slide. The next one, please. Okay, this was the ethanolic acid and the transformation products graphics. Here you can see what I have said, that the kinetic sorption is better for these transformation products. Next slide, please. In this slide, we can see that the following graph shows that the measured concentration of each compound in the soil at different depths. It is noted that positively ionized flecainide and metformin were retained in the upper layers of the soil column in the higher amounts, while sulfamethoxazole and valsartan, which are anionic in nature, were the compounds with the lowest retention. Next slide, please.
Well, can you conclude the answer? Otherwise, you not have time to questions. Thank you.
Okay. Finally, this is the conclusion of the study. Well, all the feeds obtained acceptable results. This is because they represent the relative precision. With relative precision, the delay and maximum concentration reached by the step curve. It is also determined that the sorption processes in this type of agricultural soils are determined by electrostatic sorption phenomena with the mineral phases. Therefore, positively ionized compounds have a higher affinity to sorption in the soil. Finally, the possible effects of the accumulation of substances on soil health should be studied. Further research on the natural attenuation capacity of different soil configurations and materials can be carried out for further implementation in water treatment processes. For example, the ability of this type of soil to attenuate cationically charged substances was observed here, or the decrease in the natural attenuation capacity in the absence of sufficient organic phase. Thank you. Can you please the next slide? Thank you so much.
Yeah, thank you. Yeah, thank you. Just
Ciao.
sorry if I interrupt you. It's because we have a short time for questions, and then so I want to pass to the words to Robert to deal with some questions for the speakers, please. Okay, seems like he has some problem. I don't see him here. Can you see somewhere? We have some questions here. Maybe I can read here some of them. I will start here with let's see. Yeah, I think this is to Marcela. Can the ciprofloxacin also accumulated in the crops or plants, vegetables, when they are planted on this contaminated soil? What do you think?
I have read a paper about this, and they tested ciprofloxacin on plants, and they verified that it can accumulate on plants and in different ways. They accumulate, for example, and actually it depends on the species too. For one species, it accumulates more on leaves, and other species on the roots. So it can, but it depends on the species and of course the soil type and all the other interference. I am actually gonna test it in the next months, something like this, but you know, it's just not, I cannot say it for sure because I didn't test it, just read about it.
Great. Let's see if there's another one. There's one more here. With all dangerous toxic compounds in all water, from rivers to wetlands, from lake to ocean, and high possibilities of synergic and multi-exposure component effect, it's time to pay more attention, budget or research, where we are really going on with all of this water pollution. Maybe this is a general question, if that was some of the speakers want to answer. This is a good question, in fact.
Cassiana, you want me to take a
yeah, please.
very good point. You know, in some of these compounds, we are, I would say, struggling to understand their individual toxicity. So people have always tried to go to figure out if there's synergistic mechanisms where, you know, you can add things or not, whether it's a good model to explain combined toxicity or not. And there are some efforts on it, and I would say, yes, it's something that needs to be done, but each one of these layers that we add, it's another layer of complexity on the results that we get. So there's enough budget for this thing? I will say no. You know, emergent contaminants, sadly, because there's not a regulatory framework behind them, they tend to be neglected. The only example that I can say, at least in the US, is perfluorinated compounds have come into the table lately. So there is funding for it. So we just have to keep putting pressure all over the place. So at least we get some kind of framework that will allow us to control them.
Yeah, I agree. Someone more want to complete this? Comments? Yeah, please. Hadi.
Yes, so I think I highlighted a little bit in my presentation. Also, the fact that in countries where we have no idea if these compounds exist, it's good to be proactive and really just avoid using the chemicals altogether and prevent, you know, what you see in developed countries. That was what I wanted to add. Yeah.
Yeah. There is one more question here that is interesting question. I think general one also. with so much discussion between emergent technology and nature-based solution, how to decide the best ones?
I think I can start it because it's
Yes. Yeah,
hard to find. Yeah, it's hard to find the best one because it depends on where we put the technology, or what do you need, what do we want for this technology. If you want a indirect potable reuse, a direct potable reuse, or just you want to remove this emerging contaminant from the water. So I think it depends of the location and the necessity of the place to define the best technology. Of course, you can use, for example, reverse osmosis, but it's expensive for some places to work with it. So I think we need to test in each place what's the best technology for that place. I think it's what I think about it.
Yeah, this is the challenge of the word. We can use the same solution for all of the situation, right? Someone want to comment? Hamad thanks Piero for your comments, and then he's saying that with all mega projects for wars, atomic guns, etc., then why the related UN agents does not act enough. Do you have some comments, Piero?
Sorry, can you repeat that? I couldn't. Or make a project for.
Yeah, you can see the last answer. The last question in the chat. I can read again if you.
Sure. And again, this is just my opinion. Okay, so unless we transform some of this in a real war, and again, I firmly believe that water scarcity is going to be driving a war at some point in time. There are societies that don't have enough water to sustain themselves, but we are quite good at taking the resources away. So I'm not advocating for a water war, but I guess what we are trying to do is bring awareness that these things are polluting the water everywhere, and that we're all responsible for it. And eventually we're going to end up in some kind of conflict because of not having enough water to drink.
Yeah. Okay, some more questions, comments, last comments from the speakers that you can do for this session? No? Okay, I have some all the other questions were answered and then it's published in here in our website also. Then to finalize this session, we also want to remind you all that you can see the excellent selection of submitted conference posters on our website. These posters can be found under this posters menu. And if you have some questions on these posters, you can send to the conference email, and then they will contact the poster authors and then to respond to your questions. And also with regards of the PowerPoint presentations you have been seeing, they will also be made available soon in the conference website, together with the video recording of this and the other sessions also. Some policy orientation message will be published in the IWRA UNESCO policy briefings. And we'd like to comment that the high quality selected contributions that we received will be considering for publications and the UNESCO Spring book. So may you be contacted by the group here according to that. So I would like to thank you again, all of the panelists in that session, another very interesting session to talk about emergent contaminants. and see you soon in the next sessions, right? Thank you very much for all. Bye-bye.